Hi, my name is Elisa Woods. I want to tell you a story. 2007, my son was born. He was my first child. And I'm a neuroscientist, and my husband is a biochemist. We know a lot about science, but at that time we didn't know as much about children, I'll admit. I'm the youngest of four kids. We do now. I'm the youngest of four kids, and I was never the babysitting type. I really didn't know what to expect. So when my son was born, I noticed he wasn't as interactive as I had expected. I was surprised. I thought, well, I thought babies would be a little more interactive. But then I just assumed, well, he's a baby. I guess that's how they are. I didn't know. He didn't really look at you, respond much. He sort of stared into space. He was a very floppy baby. He did some repetitive things with his hand, like this, a lot. And he was fascinated with throwing things over barriers repetitively for hours. So a lot of things like that. I didn't really think a lot of it. And then he got older. He was 18 months of age, and he started to talk. He said, "Mama," and then he stopped. And he started saying things like digga, 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 repetitive again. So it went away. It was strange. My husband started to get concerned. And I have a friend who's a pediatrician. And around 22 months, she started to say, you should get him evaluated. And the daycare provider said the same thing. So we did. We went to 5 professionals. Started with hearing, eyesight testing. That's the first thing you do. And then different behavioral professionals all the way up to the final child psychologist who gave him a diagnosis of a type of autism spectrum disorder called PDD-NOS. It's a long name, pervasive developmental disorder not otherwise specified. It's a type of autism spectrum disorder. So, when you're a parent, and I'm sure many of you are parents, I think you have a vision of the future that's clear, somewhat clear. Your child is born and you think, oh, well, they're going to develop this way and they'll go to school and, I saw him going off to college someday. And I just remember that vision became gray. It's not clear anymore. What's going to happen? So it was gray, and it was, what do we do? What are we going to do? How do we prepare him to have a happy and functional life? This is going to be something different from what we expected. So the child psychologist told us recommended we send him to ABA, that's Applied Behavioral Analysis. It's a type of therapy that's been in practice since, the 80s. And he suggested we send my son to a school, so we did. We lived in Brooklyn. We put a 23-month-old on a bus in Brooklyn and sent him off to school for 2 hours a day in a booster seat with an aide. And he'd do that for 2 hours a day, and then he'd come home for 2 hours a day and do behavioral therapy at home. And I remember before he started the school, I decided to prepare him. I was going to try to get him to learn some language. And I thought the easiest thing is identification, so I got a picture book with pictures in it, and I'd say, point to the apple, point to the house, point to the bear. And he wouldn't ever point, and when he did, it was random and he couldn't repeat it. So again, that feeling of grayness, what's going to happen? What's the future hold? I didn't know. And so after three weeks of this school, the Applied Behavioral Analysis, this intensive behavioral therapy, one of the therapists said, "Mom," because that's what they would call us, "Mom, come see what your son can do." And so I went, and I figured something small, a little improvement. 3 weeks of intensive therapy. And she said she had a picture book like the one I had been showing my son, and she said, "Show me the ball." And he showed her the ball. "Show me the house." And he showed her the house. Every single object in the book he pointed to, and I was like, this is amazing. This is a magic wand. This is exactly what we needed. And it was a magic wand for that, but I don't want to deceive people. There were other things he had to work on. So it's been 4 years of intensive behavioral therapies, and my son is currently in a mainstream kindergarten with an aide. Gone from special ed to a mainstream kindergarten with an aide. And the aid will probably be there for first grade, but I fully anticipate that he will be mainstreamed, and ultimately, I anticipate he will lose the diagnosis. Now, that's my son, and we're lucky, and I'm not trying to tell people that that would happen with any child with autism, but I do know that early and intensive intervention is critical, and that includes also identification and diagnosis of kids at an early age is critical. I've seen it firsthand. My anecdotal experience supports it, but also the research supports it. We've known since 1987, the LOVIS study showed us that early and intensive intervention provides better outcomes, the best outcomes for kids, and the biggest chance of being mainstreamed or at least having a more functional and happy life. Now, why did I tell you this story? Well, the reason I told you this story is because we're in the middle of an autism epidemic right now. There should be a slide coming up in just a moment that shows you, I want you to get an idea of what's going on. And you've probably heard it. Okay? This is the incidence of autism per 1,000 children. And you can see, this is just since 1995, it's gone up. And in 2008, it's gone up to about 11 in 1,000 kids. That's 1 in 88. And so, you know, I didn't show you the 1970s data, but it was 1 in 10,000 in 1970. And now it's 1 in 88. In fact, a study just came out from the CDC that suggests it may be as many as 1 in 50. And more boys than girls, I should also mention. So what's going on? You know, okay, you could say that better diagnosis and identification is contributing to this. Yes, I'd agree. It probably is having an effect, but not these kinds of numbers, not these kinds of increases. So what's going on, and what are we going to do about this? Well, many parents, when they're faced with a disability with their child, react by becoming an advocate, and I've done that. My husband has done that, and we're lucky because we can channel our energy into science. I'm a neuroscientist, he's a biochemist, so that's what we've done. We're looking for ways to diagnose kids early. We're looking for protein markers. So I realize my husband has these great machines that measure proteins in blood and saliva in his lab. And I'm showing you a picture. It's called a mass spectrometer. That's the machine. And this is what we're trying to do to find protein markers in the blood and saliva of kids with autism so that it's one application would be diagnosis, earlier diagnosis, and that's my passion. But there's other things we could do with it as well, this information. If we can find protein biomarkers for autism, we may also start to understand what's causing autism. We may also use it to monitor treatments. We may also use it to understand subtypes of autism, because autism is there's probably a lot of subtypes. So there's many applications for this. So we actually take blood and saliva from kids with autism, we compare it to kids without autism. And this is just a simplified diagram showing you what we're doing. We separate proteins out, we do things to treat them, to separate them out. I'm not gonna get into exactly the details right now. Before we put them in this machine, the mass spectrometer. Mass spectrometer is like, what it does is sort of akin to a detective looking at a fingerprint. It generates specific, what are called spectra. You can see that here, for proteins. That's like the protein's fingerprint. So it can tell you. You then put it in a database, because analyzing this by hand would be crazy. And you can identify the protein based on the spectra. Another important thing is you can look at the protein levels, how much of the protein is there. And if we're comparing kids without autism to kids with autism, we need to know that. We've found generally increases in many proteins, maybe as many as 100. And we're trying to figure out which ones might be important for identifying protein biomarkers in kids with autism. So, you know, I just wanted to mention this. This is the central dogma of molecular biology. It's like the rules. And many people right now, you've probably heard of the genomic revolution, the Human Genome Project. Most people are looking at genes. I'm not saying it's bad to look at genes, and we certainly have plenty of evidence that there are genes involved in autism, probably in collaboration with environmental factors. So it's a gene environment problem. That's likely. from what we know, and many genes, not just a few. Again, those autism subtypes. That's probably what's going on. But we have so many questions that still need to be answered. So most people are looking at genes. That's DNA and RNA. We're looking at proteins, and very few people are. And we think that's important. So DNA makes RNA, makes protein in a cell. Proteins go out and do things, which is why we're focusing on them, because we think they're the functional unit. So they're the thing to measure. They're probably the thing that can be changing, maybe with environment, for example, maybe with pollution. We don't know. We need to keep looking. We've actually found some things in blood and saliva. I'm just showing you 3 of the proteins we find interesting. This is just showing you 7 kids versus 7 kids. Now that's not a lot, and I'm not going to pretend it is. It's what you'd call pilot data. We need funding. We need to look at more kids. So that's something we're working on, trying to get funding from different agencies, government agencies, foundations, et cetera, to look at more kids. That's kind of the limiting factor right now. But 7 versus 7 kids, we have identified some things that may be interesting. SPA1, for example, that's a short name for this protein, increases when people are exposed to pesticides. So I think that's an interesting one. This is a cholesterol regulating protein. And this is a type of hormone regulating protein. So we could start to look at these proteins and figure out maybe what's going on. And they also probably interact with each other. So it's what we call a protein biomarker signature. I think anyone can understand this graph if I explain it to you. You don't have to be a scientist. So these are proteins. So the previous one was blood. These are proteins we found in saliva. Each oval is a child with autism. We measured the proteins 3 times. Each rectangle is a control, somebody without autism. These are just dysregulated proteins without even knowing what they are. Look at how different they are. And this is what we found in saliva. So I just saw this, I was amazed. It was more than I expected, but it's so clear to me. There's something going on, and there's something we can measure. This could form the basis of a diagnostic test, which could get kids identified earlier and treated earlier. And from my story, I know how important that is. So we want to move forward with this. Again, study more kids, and we are in the middle of an autism epidemic. We don't know why. We need to be asking these questions, and I think these are questions worth pursuing. Thank you very much.